So, ladies and gentlemen, welcome to the afternoon session. We have again two distinguished speakers for this afternoon. As you will see from their CVs, they actually have many things in common, although their titles and professions are rather different in a certain sense. So let's start with the first speaker. She's actually a chemist, and we asked what should a chemist have to say about, well, this is quantum coherence, but actually quantum physics is getting more and more interdisciplinary, and chemistry is quantum physics. And Birgitta Whaley is from the University of Berkeley. She got a degree in the University of Chicago, had also a time in Israel, so that's something that connects her also to Moti Haibel, who's now in Israel. She was at the Hebrew University in Jerusalem and at Tel Aviv University as a postdoc, and has been a professor now already for quite a while at the University of Berkeley. And her specialty is, well, it's hard to say it's a specialty because her field of research is so broad. She has papers in condensed matter systems, including helium nanodroplets and atomic physics, molecular physics, but also in quantum information processing. And so that combines nicely to a field which connects molecules and quantum information, and that's in quantum coherence and entanglement in biology. And we're happy that you're here, and looking forward to your talk.
Okay, so first of all, I'd like to thank Anton and the other organizers very much for inviting me to this very interesting and wonderful symposium, and it's a great honor. I regard it as a great honor to be able to talk to you about not only our work, but I'm going to present an overview of current work which examines particular quantum coherent effects in biology. And what I'd like to do is to show you in the beginning part of the talk how far Schrödinger got with his thoughts about biology, and then show you how what we're all doing in the current time takes off from that and continues his ideas in this area. And specifically the work from our group that I'll present is work, so my group is a theoretical group, so it's work from a sort of pretty interdisciplinary group of physics, chemistry, and quantum information people. And we're doing this together with the group, experimental group of Graham Fleming, who is, as you'll see, is one of the founders of the experimental investigation of quantum coherent effects in biology, and with two experimentalists in his group. And it's actually funded by DARPA, because the original goal of this specific collaboration was to investigate whether there was any quantum information processing evident in these coherence effects. And I'll tell you about that story as well. Okay, so first of all, some comments on Schrödinger and his relation to biology. So these are the pertinent publications for what's of interest in biology of quantum mechanics today. I prefer not to use the term quantum biology because it's a little bit loaded. But basically, as we heard this morning, there was this wonderful year, 1926, when he published the first total of 6 papers on wave mechanics. And then for our purposes, also 1935 is an important year from Schrödinger's perspective, because he coined the term entanglement for the Einstein-Podolsky-Rosen paradox. And first of all, he had a paper in German where he called it Verschränkung, and then he wrote a paper in English, the term entanglement basically is stuck. Then in 1943, he discusses the relation of physics to biology in his Dublin Lectures. Notice the subtitle of these lectures. The title is normally referred to What is Life. The subtitle was The Physical Aspect of the Living Cell, and that is indeed what he focused on in detail. Then in 1956, he gave another series of lectures which focused exclusively on the relationship between mind and matter. And these are, I'll say more in detail about the 1943 lectures. I'll say a little bit about the 1956 lectures, but less. Okay, so the 1943 lectures Okay, the most important pieces that I take from these lectures are the first two here. Firstly, and I've used here some of his terms, which are somewhat out of date, mixed with some modern terms. I used the word permanence, which he used a lot. The permanence of the genetic material is due to the quantum mechanical structure and energetics of molecules. And this relied heavily, or this was really relating very much to the pioneering work of Max Delbruck and others. And secondly, another major point that he made was that the energy gaps between molecular quantum states and also potential barriers between the different states gives rise to a very rare instance of fluctuations, and that conveys a stability to what he called the heredity mechanism, meaning the genetic system. So these were the two important aspects that he pulled out of the situation in understanding of molecular biology as it was at that time. He also had quite a bit of discussion about the dichotomy between thermal disorder, or statistical physics, and what he called order, or implicitly quantum order in biology, namely the energy gaps and energy structures were basically what was giving the order. And again, referring to the fact that in genetics, events that happen are a paragon of orderliness. So he was basically arguing that the order imposed by the quantum structure of the molecules was responsible for the ordered nature of the genetic process, and in contradistinction to the kind of thermal fluctuations and thermal disorder which one might expect from classical statistical physics. He also made the point that there was more to be understood. In particular, he said that this molecular understanding of the genetic stability doesn't give any insight into the functioning of genetic material. And indeed, it's rather poignant that his lectures were published, or his lecture was written in 1943 and published in 1944, and DNA was first discovered in 1944. So this goes to one of the areas where he's been criticized for, because he didn't really quite have the right understanding of the structure of a gene. He thought of it as a single large molecule and didn't appreciate the DNA encoding in it. Now, I don't want to go into more detail about what was right and what was wrong here and where it came from. You heard this morning from Professor Thiering that it was really quite and I agree that it was a very reasonable presentation of the state of the field at that time. And it did rely heavily on other people's work. I don't think he claimed to be very original in any of it at all. And it was really, what's interesting to me was to go back and look, especially knowing a little bit about the history of chemistry that I do, but I don't know much about the history of biology. But if I look back to what had come before, on what was he basing the knowledge which he put into that little book. And that's quite interesting to look at this, because there were some things that were missing that could have been there at the time. So if we consider from a little bit before wave mechanics, the notion of a potential energy surface, which is critical to the arguments of stability that he puts in the book. The notion of the potential energy surface was first basically put forward by Marsden in 1914. Marsden was unfortunately killed in World War I, so I didn't hear from him beyond a few quite powerful papers expressing the notion of phase space going over a potential energy surface, and the notion of the activated complex, making it more mathematical than it had been in the Arrhenius formulation. So then we have the Schrödinger wave mechanics, Heisenberg Dirac, the resonance and exchange phenomena, and this all, from a chemical point of view, this all proceeded very, very fast. From the wave mechanics all the way to the full understanding of the chemical bond was basically a year or two at the most. The London theory, the valence bond theory, came in 1927, and then Hund-Mulliken, the more powerful molecular orbital theory, a year later. Also in 1928, Gamow identified the tunneling phenomena. And I put it down here, 1933, the Journal of Chemical Physics was established, because by then the whole field of application of quantum mechanics to chemicals had become so large that it deserved its own journal separate from Physical Review. 1933, also Bell did calculations showing that there's a possibility of hydrogen tunneling in chemical reactions. And then 1935 was a big year all around. There was the EPR paper on local quantum correlations, Schrödinger's papers about summarizing the state of quantum mechanics at that time, identifying the entanglement, which he there said was the characteristic traits of quantum mechanics, that which would make it really distinct, different from classical mechanics. In the same year, Max Delbruck and his group published a seminal paper on mutation and genetic structure, which is the source of much of the material in his book, in Schrödinger's book. Also in 1935, and Schrödinger seems to have been less aware of this, the theory of chemical reaction rates was really put on a firm quantum statistical footing by Eyring and Evans-Polanyi, based on many, many people's work since 1914 in Marcelin. And then in 1943 with his live lectures. So in the light of this, it's really, everything he writes in the book is really quite reasonable. And it's maybe a little bit surprising that he didn't make any mention of tunneling. But at that time, tunneling, like also the quantum coherences that I'm going to talk about in the second part of the talk, had no experimental manifestation in experiments, biological experiments at the time. Okay, so that's what was in the book. Now, the last, then the second book goes to Mind and Matter, and I just want to make two comments here. At the end of the 1943 lectures, there's a statement on Schrödinger's text which says that quantum indeterminacy plays no biologically relevant role in, and then this is more or less, not quite a quote, in bodily events, or he said, I think, time and space, bodily events corresponding to activity of the mind, except possibly by enhancing by the accidental nature of meiosis, which is cell division, mutations, etc. So Schrödinger was making an explicit statement about the relevance of the non-trivial parts of quantum mechanics to the mind-matter division. But he didn't make any comment about whether quantum indeterminacy might play a role in the more mundane aspects of cellular function. And that's really what the current experimentation in the area is really focused on, not on this mind-body, on the interaction of the quantum indeterminacy with the mind-body. In his 1956 book, Mind and Matter, he goes into this in more detail, and in particular, the main focus of the book is can you isolate and can you determine which are the material processes associated with consciousness. He also spends quite a bit of time talking about what he calls the mystery of the sensual qualities, namely how it is that we perceive color and different colors or tone. And it's not one of the main things in the book, but I put it here because I want to indicate a link of some of the current experiments to this particular aspect of his interests. Okay, so that was then, and this is now. So now, if we look at what people are doing in investigating the effects of quantum mechanics in biology, it's no longer concerned with the consequences of energetics of molecules, the fact that we have discrete molecular states and that we have energy barriers to get over. It's actually more directly concerned with the dynamics of how those processes happen. And as we know from the many years of chemical reaction rates, once we've constructed a potential surface, we could still then approximate the dynamics as completely classical, or we could incorporate quantum effects. And this is a table which describes basically the main phenomena that are being investigated today in terms of quantum dynamics. The phenomena are shown here. So photosynthesis, primary steps, which I'll talk about. The focus of the second half of the talk will be this. Vision, There's also been a lot of work done on excited states in vision. Then enzyme catalysis, photosynthesis, and bird navigation, this refers to the putative mechanism by which birds can detect the magnetic compass of the Earth to navigate when they go on their long migrations. The quantum processes which are involved in these or believed to be involved in these biological phenomena with varying degrees of authentication are, for the photosynthesis, energy transfer and electron transfer. In vision, it's a very important critical photoisomerization process. In the enzyme catalysis and photosynthesis, it's the electron tunneling and hydrogen atom transfer in enzyme catalysis, proton-coupled electron transfer. And in the bird navigation, it's essentially a radical pair mechanism which has been proposed and for which there's a lot of evidence, but it's not conclusive. So if we're looking at this table now in a little bit more detail, we divide up the experiment. So now we're saying from wave mechanics to biology after Schrödinger's seminal book. So there was still a big gap before there were any real experiments, but in 1960s they started to come. And the first was a demonstration that photoisomerization is the primary step of vision. And that's been then, since then, followed by many, many studies on the dynamics. Femtosecond dynamics have been identified, and they show vibrational coherences. And most recently, it's been shown that this photoisomerization proceeds via a conical intersection. So that involves very intimate complicated mixing of electronic states and really a lot of changes in electron correlation. Then in 1966, as far as I could trace it, this was the first experimental demonstration of electron tunneling in photosynthesis. 1989, 20 years later, hydrogen tunneling was demonstrated by a colleague of mine, Judith Klinman, in enzyme reactions. And there's quite a few studies on this now. And then in the current century, 2007 and onwards, starting with Graham Fleming's group's experiments, it was demonstrated that electronic energy transfer in light harvesting complexes, which are the first step in the photosynthetic cycle, that this electronic energy transfer is accompanied by quantum coherence. And this was first shown at low temperatures. And then last year, two former postdocs of Graham Fleming, who are both independent now, Greg Engel and Greg Scholes, with their own groups, they both showed in different systems that this quantum coherence persists at ambient temperatures. So this now raises a really fascinating question as to multiple questions. Firstly, whether this these experiments, of course, are done in vitro. The first question is, which we believe is positive, that the quantum coherence, whether it exists in vivo. And secondly, the bigger question, whether or not this quantum coherence plays any functional role for the light harvesting. So these are the systems where quantum effects have been demonstrated experimentally. Then in addition, there's been many proposals, and starting with possibly what would be the one that Schrödinger would have immediately dismissed, the Hameroff-Penrose proposal that there's quantum coherence in brain microtubules, leading to superpositions. And there's no, so I put these here because distinct from the experiments, there is no experimental evidence for any of these to date. But the 1996 Turing proposal that olfaction, namely our sense of smell, occurs by inelastic electron tunneling, which gives it the sensitivity to molecules characterized by specific vibrational states. There's been quite a lot of studies which showing that the sense of smell, certainly in Drosophila, is consistent with this, although there's no direct proof that there's tunneling as to date. The 1998 Klaus Schulten and co-workers proposed that the bird navigation, the magnetic compass scheme of birds, relies on a radical pair mechanism. And while there's been no conclusive experiment showing, yes, it is indeed this, and it's not small particles of magnetite in the bird's beak, which is another option, there have been many, many experiments which are very much consistent with this for about 4 or 5 different types of birds. So this is really growing experimental evidence that this is probably at least one contributing mechanism. And then last year, there was a proposal from Aliphashi Vaziri, who I believe is joining the faculty here in a couple of months, and Martin Plenio, that there would be also some similar kind of quantum coherence, meaning superposition of configurations for the transport of potassium ions through ion channels. And this is, again, at the moment, a completely theoretical proposal, and maybe Vaziri will be doing some experiments on this in the next few years, maybe. So I put an asterisk by these 2 points here. One, the experiments on vision, photoisomerization, and secondly, on electron tunneling and olfaction. I put the asterisk there to remind me to point out to you that these are both systems which go towards the mystery of sensory perception that interested Schrödinger so much. Although it's doubtful that either of these establishing the way the primary events in either of these sensory processes would actually satisfy Schrödinger's interest, it's a first step on a very, very long and possibly infinite road to understanding how we sense things. So this is a slide which Ali Pasha put together and kindly loaned to me, showing the current state, at least of the dreams, of those of us interested in understanding large-scale biological function through the eyes of quantum mechanics, showing the typical time scales up here and the length scales on the x-axis, showing the division between where we normally typically put quantum at small length scales and short time scales, classical at large length scales and large time scales, and showing our tools are going in this direction from large scale, going down to ever better precision in space and time, which allows us to go into this regime. And also in terms of function, we're seeking always to understand at a more molecular level, at a smaller and smaller level, how these very complex biological systems work. And one of the immediate questions when one's in this regime down here is, can the quantum coherence, which is characteristic of quantum superpositions and goes beyond the internal structure of atoms and molecules, can quantum coherence of electrons, for instance, in different units, be relevant for the biological function? So that's one of the grand goals of this area. So now for the rest of the talk, I'm going to focus on electronic energy transport. This is the most extensively studied non-trivial quantum effect in biological systems to date, and that's largely driven, I should say, by experiments, because there are so many experiments now. I'll show you a summary in a moment. So what is the background of this? Well, until 2007, electronic energy transfer in biological systems, including light harvesting systems, was always described by the Förster theory from 1949, which is basically a theory of incoherent hopping, which you can derive by making basically a golden rule calculation for the transfer of energy from one chromophore to another, which is governed by a dipole-dipole interaction. And this has this classic distance signature of 1 over r to the 6 coming from the dipole-dipole interaction. So in the experiments of Fleming and co-workers came out in 2007 showing that there was some coherence in the transfer of energy between different sites. The question was how, firstly, how does it play a role? And secondly, why? And what I'm going to show you is some studies that we've made of the following three aspects. Firstly, in terms of how it happens, we first of all have to understand how we get quantum coherence in an open quantum system. So I'm going to show you what's involved in simulating this, or getting to the point of having a reasonably accurate, we believe, a reasonably accurate simulation of the quantum dynamics. Then I'm going to answer the question, or address the question and answer it, whether this transport shows any kind of quantum speed-up as necessary for performing a quantum algorithm. So this is where our motivation coming from quantum information theory drove us. And lastly, I'll talk about whether this coherence is accompanied by entanglement and what that means. Okay, so I imagine that most people here, or many people, are not familiar with the details of photosynthesis. So let me just put in context of the entire photosynthetic system what the light harvesting, where the light harvesting step is. And it's actually right up here. So on the left here, there's a schematic. So the light is absorbed, and just right at the top, the point where the light is absorbed here, and then transferred across some structure, which is called the light harvesting complex, to the reaction complex. There it causes, the excitation energy causes charge separation, and then this is followed by chemistry, and these reactions here, which then build up energy in the organism. So the important features of this light transfer here, and so here actually this picture indicates the hopping nature, which was understood to be the nature of this energy transport from one chromophore to another, or until about 2006 or 7. And this process is famously efficient. Its quantum efficiency is actually about 99%. Its energy efficiency is less, but the quantum efficiency, meaning for each photon absorbed, you get one excitation out going to the reaction center, is incredibly efficient. And the other important characteristic is that there's a kind of energy funnel here. So the light is absorbed by the higher absorbing, so for instance, blue absorbing pigments, and then it's getting transferred to lower and lower energy until it gets to the reaction center. And in the reaction center, that's where all the chemistry happens, but we're not going to address any of that. So we're only going to be interested in what makes these light harvesting systems operate in a quantum coherent fashion. Well, if you start to look at these, and whether you're a chemist or a physicist, if you start to look at them, I mean, if you're doing sort of quantum dynamics, and you say, well, I'm going to look at one of these, I'm going to study one of these, and then someone shows you a picture of what they look like, you sort of, first of all, think you might pack up and go home, because they're incredibly complicated. Up here, I'll show you better a picture of this later. This is actually one of the more attractive looking ones because it has a high degree of symmetry. This is the LH1 and LH2 in purple bacteria, which has sort of two ring structures. You'll see them more later. This is the system which is found in 50% of green matter on Earth, PS2. So it's a very, very important system. And this has about 96 different chromophores in it. And the basic unit here is something called LHC2, which consists of a trimer. And for reasons not entirely understood, many of these light harvesting complexes do have a trimeric form. That might just be a redundancy feature. So in each trimer, there are 14 chromophores. So there's a very large number of chromophores. And the next thing is that these chromophores, so these, in this case, these are the blue objects behind these gray swirls, the gray tendrils here, which are proteins which pack these chromophores tightly together. And these molecular aggregates, the chromophoric molecules, are often, but not always, to add more complication, they're not always embodied in protein scaffolds, but they often are. So, and this is just, this is a little bit dark, but this is just to show you actually a very beautiful picture from Klaus Schulten's group. This is a simulation of a vesicle which they believe is formed by, on the outside of cells. So you basically have this sort of spherical type of object formed on the outside of the cell, which contains about 1,500 chlorophyll molecules, basically many, many of these rings of LH1 and LH2, with some deformation of the surface. And the reaction centers are actually these blue things located inside the larger rings there. Okay, so we're faced with this very beautiful but very complex structure. It's really a challenge to imagine that there might be quantum effects other than the basic electronic structure of the molecules. And is there entanglement, or is there any enhanced, quantum enhanced energy transport? So those are the basic questions we wish to understand. And of course, the way we're going to understand them is not by immediately attacking these very complicated systems. We look instead at a very simple system. The most simplest system, and also the most extensively studied from a theoretical and experimental perspective, is the light harvesting apparatus of green sulfur bacteria, which lives in these swampy environments. That's the green sulfur bacterium. Doesn't look very nice. This is its photosynthetic light harvesting apparatus here. And these are the objects which absorb the light. So these are actually the light harvesting antennas. And what we're going to look at is this object down here, which is called the Fenner-Matthews-Olsen protein, which is a light harvesting system, but it doesn't actually directly receive sunlight. It just transmits the excitation energy from the antennas through the cytoplasm to the reaction center. So this object actually receives the excitation already as an electronic excitation rather than directly as a photon. But its function is otherwise, its mechanism is believed to be the same as that of a chlorosome up here. Okay, so this is the structure of this FMO system. You see again it has a trimeric structure, and the chlorophores are these red structures hidden behind the blue and the green, which indicate the protein scaffold. And this has been the entire structure of this has been very well characterized by X-ray crystallography. This is another view of it where there's no distinction now between the tendrils and the sheets of the protein, and now you have in green here the chloroplasts. And if we strip away the chloroplasts, you see here this is the basic structure of the chlorophores. So there are 7 chlorophores here, and each of them is a bacterial chlorophyll molecule. They're actually all identical, 7 identical chlorophyll A molecules, and what's shown here are just essentially the organic aromatic part of the molecule, which is a ring like this, ring structure, and then there's a long tail which tethers it in the protein scaffold. That's not really shown. So the electronic absorption occurs from the ground to the excited states of these molecular systems, 7 of them, and the electronic state of this system has been very well studied. There are several very good Hamiltonians for this system, and if you take one of those Hamiltonians and you diagonalize it, you'll see that the excitonic structure now. So if you imagine one excitation being put onto this complex, one finds that there are 7 excitons, and these are the energy representation. These excitonic states are mostly delocalized on two bacterial chlorophylls. Okay? So we don't have a band of excitations. We don't have states which are, we don't have a band of excitations. We have rather more disparate pairs of delocalized excitons. And that's the fact that these are disparate, it's not a band, is due to the packing, which we can characterize, but we don't understand why it's packed in this particular way. This is part of the open questions I'll come to at the end. Now, experimentally, this system's been very well studied by pump probe and all kinds of spectroscopic experiments before the ones that indicated coherence. And so this is a summary of what the timescales involved are. So if we consider the FMO here, which transmits energy to the reaction complex, the energy is absorbed on this side, 1 and 6, remember those numbers, and it has to get transferred to the site number 3, which is connected to the reaction center. So this is a realistic picture here. And the excitation starts sort of like this, if it's absorbing light from here or excitation from here. And then within 5 picoseconds, 4 or 5 picoseconds, it's basically exclusively on the site number 3, which we refer to as a trap, which is connected to the reaction center. So everything is over in a few picoseconds. And that's, again, that's actually a similar time scale to that involved in the primary step of vision. So the way the coherences were investigated was by making a two-dimensional femtosecond spectroscopy, which is a third-order nonlinear spectroscopic technique where one sends in 3 phase-matched pulses and measures at some phase-matched condition with time delay t. And so one's measuring here the output electric field, which is directly proportional to the polarization of the system. And this gives one then a response function which tells you about correlations between the absorption frequency in here, the omega 1, and the emission frequency, omega 2, as a function of what we call the population time, t, which is the time between the second and the third pulses. So it retrieves this correlation. Now, if you're not familiar with this technique, think of it like a generalized spin echo technique. If you remember the spin echo, the Hahn spin echo, you excite, you're starting off, say, in the North Pole, you go down to the equator, the dynamics happens in the equator, and then you reverse, and you refocus, and come back up again. So in this case, you would be going down from the North Pole to the equator. You have some dynamics, which happens. You put a second pulse in, which takes you not back up again, but it takes you down to the other end. So you're going to what would be the excited state. And if you have more than one excited state here, then you have an opportunity to see coherences, or the quantum evolution of those multiple excited states. And when you then come back with a third pulse, and essentially the third pulse does the refocusing, the signal you see then has an imprint of the coherences during this evolution time. You won't have any of those coherences for a 2-level system, but if you have a multi-level system, you'll see them directly. So this is actually what's measured. So schematically, so the signal is a function of these 3 times, and basically there's a Fourier transform with respect to the first time, and this is done directly, so you don't have to Fourier transform here, but the result is that you get a spectrum as a function of 2 frequencies, an excitation frequency and an emission frequency, excitation frequency here, an emission frequency, and you see structures here on this 2-dimensional peak, on this 2-dimensional spectrum, and those structures tell you about what processes are happening coherently during the time capital T, in which you're basically in the manifold of excited states. So this is one of the first measurements that was made. This is for a particular population time, T is 200. This is 2D electronic spectrum in this FMO complex in vitro. So this is a large set of samples of molecules, all in solution. And if one takes a diagonal, if one takes this measurement and makes it for many different values of capital T and puts these together, then you get this 2-dimensional electronic spectrum as a function of time. And this shows now these oscillations here, both along the diagonal, and you also see here, off diagonal, there are some, this is on the diagonal, so this is now a little peak way down here, which doesn't show up there, but you see there are also some oscillations here. So on the diagonal, it tells you about the populations. And this particular peak here reflects the lowest exciton, exciton 1. And this is a beating signal that's extracted from this. It's Fourier interpolated. And I should say this is the very first data that was obtained in 2007. Now there's a great deal more data. There's different ways to obtain the data. And so there's the Fourier interpolation is really not in doubt at this point. And if one then Fourier transforms this, one gets a power spectrum, which is the dotted line here. And this power spectrum was compared with the expectations of the predicted beat spectrum, which are obtained from the excitonic structure of the complex. And see, there's very good agreement. So this was the basis for which in 2007, the basis on which the claim was made that the oscillatory signal seen in the 2D electronic spectra is consistent with electronic coherence. So the electronic populations are stabilizing, but the coherence persists. And this was taken as evidence for coherent wave-like energy transfer of the electronic excitation. And these kind of oscillations are also seen if you go off diagonal. There's small peaks that you can follow. So that was the first experiment. And so that basically said that this picture of incoherent hopping is either completely wrong or is gravely in doubt. And that was at 77 Kelvin. So since then, as I indicated before, last year, there's been experiments done on this particular system, again, this is room temperature in Chicago. It's not quite the same as room temperature in Berkeley, but maybe approximate the room temperature, like here, yeah, right. Right, and then, let's see, and then the other one was Toronto, it's even worse, right? So the other one was a completely different system. These are phycobility proteins in marine algae. They have somewhat different structures. And the average distance between chromophores in this system is about 20 angstrom. The average distance here is about 10 or 15 angstrom. So this is a somewhat more extended system. And they also see these oscillations, again, at room temperature. Then other systems, so these are quite well established at this point. And then in the LH1, LH2, which is another bacterial system, there have been many, many studies, which not so directly, there's no two-dimensional spectroscopy, but there are many studies that indirectly indicate that there are oscillations, coherences, populations going around these rings that I showed you. And a third one is in the reaction center itself. This is from the Fleming group. They did a femtosecond time-resolved study of the dynamics of the excitation in the reaction center, and they also found coherences. And then most recently, 2 years ago, the same experiments were repeated on LHC2, which is this larger system with 14 chromophores, and similar coherences were found. So these are, there's really now, I think, a large body of experimental evidence that the coherences, electronic coherences here, and that they're real. Now this, I'm just putting this up again to show you that in the purple bacteria, that's this system with the beautiful rings, one might not find this so surprising because the structure here is very symmetrical. So around these rings here, this is LH2, LH1. So around these, there are 18 chromophores which fall into groups of 9 pairs. And they all are pretty much in equivalent positions. They all have pretty much the same energies. And they're equivalent. So it's not so surprising that there might be coherence as in the way the electronic excitation might be delocalized over these rings. But in the other system, in the FMO, where all the sites where this energy funnel is really quite strong, it's actually quite surprising. So now I come to the theoretical analysis of these dynamics. So the question we are asking is, does this reflect some non-trivial quantum behavior, where we're saying that anything that involves just the fact that molecules have energy levels, this is trivial or well understood? So there's 2 things that we're interested in. One is entanglement. And so while there's been many, many studies, and you'll hear more about them later on this afternoon, about entanglement in solid-state systems, ordered systems, relatively ordered systems, at low temperature. This is, for instance, an analysis of the susceptibility of a magnetic material based on entangled pairs of spins. In biological systems, however, at physiological temperatures, these are hot, wet, what we would call noisy systems, And it's not at all clear that there would be any kind of entanglement there at all. And so really, whether there is entanglement or not is a sense of watermark for whether our classical, or let's call it quasi-classical, with the underpinnings of molecular structure, a description of biological reality could be considered complete. And the second point was quantum information processing. So the moment this experiment was done in 2007, there was a lot of buzz about plants performing quantum computation, based on the notion that because you had this energy funnel, and there was also a spatial distribution of chromophores, that the energy had to search for the exit to the reaction center, and so the plant was performing some kind of quantum search algorithm. So to test that, we looked at whether there was, that was motivated us to look at quantum speedup. So firstly, let me say a few words about first of all, simulating this system theoretically. I won't go into great detail, but just to give you an idea what's involved. So this is a prototypical light harvesting complex. So if you can do it for these systems, I mean, it shows the same kind of features that all of them do. So we have these Frankel excitons on these molecular chromophores here. So in this particular system, the energy levels are very well known from pump probe experiments and theoretical modeling. And so we can write down a fairly, with confidence, a Hamiltonian for the closed system dynamics just of the set of 7 chromophores, consisting of the site energies and the dipole coupling matrix elements, which have been calculated and also to some extent measured. But we have to take into account this protein scaffold surrounding the system. So it's an open quantum system. And when we look at what's involved with the protein scaffold, we see that we have 2 problems. Firstly, the coupling of the chromophores to the bath is pretty large, and it's not bad that it's large. What's bad is that this coupling is exactly the same order of magnitude of the electronic couplings here. And furthermore, the bath is not a Markovian bath. There's a well-established relaxation time, which corresponds to energetically, guess what? The same energy scale as the electron-phonon coupling and the electronic energy transfer. And all of these timescales are faster than the reaction center trapping rate. So fortunately, we don't need to take much account of this. So this means that we have three timescales which are the same. And so this is just a quick note. We're analyzing the experiments. We use a density matrix because that's what's appropriate to the in vitro situation, where one has basically a large ensemble of systems, at least today. In the future, we hope that single molecule experiments will be possible. But so here one has a system where we have 3 timescales which are the same. And it's quite hard to simulate the quantum dynamics in this situation. So what Aki Ishizaki and Graham Fleming have done, so in particular Aki Ishizaki has developed a non-perturbative, non-Markovian treatment which relies on assumptions of Gaussian fluctuations and linear coupling. And certainly the Gaussian fluctuations is a very good assumption here. And it's basically a hierarchical expansion of a time non-local set of equations. And it has a property, most important property, it preserves the fluctuation dissipation relation, and it does correctly take account of the temperature. So that's all I'll say about that because I don't have time. But this is a result from Aki Ishizaki's work showing and these are the most realistic simulations of these systems to date, showing in this case the populations as a function of time for the 7 different sites, when we have initial excitation either on site 1 or on site 6, or on 300 Kelvin now on site 1 at site 6. And you see these oscillations even at the room temperature calculations, which indicate that one has these coherences. Now, I should say that the initial conditions here being initial excitation either on site 1 or site 6. So all the calculations that are done are designed to emulate the in vivo conditions where one only has 1 photon entering the system at a time, rather than the broadband excitation in the experiments. Okay, so now first of all I'll tell you about how we analyze these dynamics to determine whether or not there's entanglement. And so this is a basic summary. This is the Bohm analysis of the EPR paradox in terms of a pair of coupled spins, which we're all familiar with. And we know that if that the measurements done at one end the results of those measurements depend on which measurement was done at the other end. So these are the non-local quantum correlations, which are an essential part of structure of quantum mechanics. As we had a very nice analysis this morning from Professor Fröhlich about this. And this is, again, it's a reminder of Schrödinger's view of this. And also pointing out, you'll hear more about this from Anton Zeilinger, I'm sure, that the quantum information perspective on entanglement is that it's a resource. It's been used to advantage in quantum communication. It allows teleportation, key distribution, and it's also been used in quantum computation. It enables some quantum algorithms, measurement-based quantum computation, and so on. So it's a key resource for information processing. It also, and I think you'll hear about this from the third talk this afternoon, it also provides, in recent years, provided insights into strongly correlated quantum materials. So it's determining whether it exists or not in these systems is quite interesting for many reasons. And while you all know this, I wanted to present this again because this is a very nice summing up by Schrödinger of what entanglement is. If one looks at a two-particle pure state and just looks at the von Neumann entropy as a measure of entanglement, and you then look at the reduced density matrix of any one of those particles, you see that the entropy increases, and this is a signature of these two subsystems being entangled. And again, Schrödinger puts it very, very succinctly with the statement that the best possible knowledge of the whole, which in other words, the knowledge of the entire two-particle wave function, does not include with zero entropy, does not include the best possible knowledge of its parts. So knowledge of its parts is then incomplete. Okay, it's a very nice statement. Okay, so how do we classify entanglement in these light harvesting systems? Well, unfortunately, we don't have a pure state. We have mixed states, we have finite temperatures, and we also have multipartite systems. We don't have two nice, well-separated spins. We have 7 chromophores. We do, however, have a great simplification, which is that we're looking at a single excitation subspace in FMO, and these are indeed the in vivo conditions for FMO. The number of light photons incident are very, very small. In some instances, below the ocean, it's about 1 per half an hour. So we're considering a single excitation entering the complex from the base plate. So the analog of this from optics would be mode entanglement. I'll come back to that later. So given these simplifications, we managed to actually construct a tractable multipartite mixed state measure that we could actually compute from the simulations, which is based on the relative entropy of entanglement. And then if you look at this in detail, you see that the witness for entanglement here, the quantity which tells you whether or not there's any entanglement at all, is just the sum of the off-diagonal elements of the matrix of the coherences squared. So this tells us that the entanglement in this system is essentially equivalent to the quantum coherence, this mode entanglement. Secondly, we looked at bipartite entanglement measures. The standard measure for this is the concurrence. That's pretty easy to derive. And these are the results. On the left is the global entanglement for the FMO complex. On the right is the chromophore bipartite entanglement for different pairs. And so what we see, so we're basically analyzing these simulations obtained with a reduced hierarchy approach. And we see that at short times, so up to 100 femtoseconds, there's large amounts of both global and bipartite entanglement generated, not surprisingly. We have now, in this short time period, up to 100 femtoseconds, we're having transfer to the neighboring chromophores. But the important feature for us in terms of biological action here is that there's significant entanglement at long times, steady state. So we're basically seeing steady state entanglement, which is limited only by the trapping dynamics, which happens on a longer time scale here. And this is significant. This maximum entanglement here is log 7. It's about 2 point something. So this is about 15 to 20% of the maximum possible. And this global entanglement is accompanied by bipartite entanglement across multiple partitions. In particular, remember I told you that the trap is site number 3. Well, site numbers 1 and 3 are separated by about 30 angstroms. That's the largest distance across the complex. They're definitely not bonded, not even van der Waals bonded chromophores. And we see that if we start with initial excitation on site 1, we have a very rapid rise of concurrence with site 2, but we also have a rapid rise with concurrence with site 3. So we're building up this bipartite entanglement across the entire complex, and this decays very, very slowly. Again, appears to be a more or less steady state value. So we conclude that there's long-range entanglement, where long-range means about 28 angstroms under the in vivo conditions. And these are the kind of distance scales that are involved also in the bird navigation system. I don't really have much time to say a lot more about that, but that radical pair mechanism relies on the formation of a spin pair, singlet spin pair, which is separated by about the same distance, 28, maybe up to about 40 angstroms in some conditions. And then the question in those studies is whether the entanglement of the singlet, whether that significantly enhances the sensitivity of the system to a magnetic field. Okay, so this entanglement, there have been many other studies since then. Since our initial study, we're using different measures. Some of you may know there are many, many different measures of entanglement. And as long as one has a calculation which has the right realistic temperature dependence, then basically one gets very similar results. Also with these different systems, so the larger system, LHC2, with 14 chlorophylls, so of 2 different types here. Ishizaki's looked at his simulations for these, and he finds also using a different measure of entanglement, he has, again, some steady state entanglement between the 2 different types of molecules. So it seems to be quite generic, this mode entanglement of the systems. Okay, so how much time's left?
Nearly finished.
I'm nearly finished. Okay, so maybe I'll just go to the conclusion of the speed up thing. I'll say one more. Okay, so I'll do that, and I'll come back to the entanglement when I sum up. Okay, so the second thing that we looked at was, is there a quantum search? So remember I told you that half of the green stuff on Earth is photosystem 2. So if this is natural quantum computation, then this would be really very interesting for many reasons. So what we did was we took the quantum walk approach, which we know from previous work that we've done can give us a quantum search algorithm, and we adapted it to the system where we have now not the nice energy, the flat energy landscape of a maze where we normally visualize in information science how a spatial search would proceed. But now we have this disordered random environment, so we also have these energy funnels, which would be the underlying ordered structure. And in addition, we have decoherence. So then we ask, can we still get speed up of the type needed to perform a quantum computation? So the procedure we took was very straightforward in a way. We looked at the Hamiltonian matrix representing these 7 chromophores, and that's shown here, subject to about 12,000 wave number shift relative to the ground state. So these are the 7 excited electronic energy levels in the site basis. And we look at the largest elements in that matrix, and that defines for us a pathway through the complex. And this pathway turns out to be rather strongly one-dimensional. And so then we look at the evolution of the electronic excitation as it goes through this system and compute the mean square deviation, which allows us to distinguish between a classical random walk, which would be this diffusive spreading here, where x squared is proportional to t, and the quantum random walk, which has this characteristic behavior with ballistic type spreading. Okay? And we know from the work on quantum algorithms that you need to have this underlying physical speed up in order to do a spatial search as a quantum with speed up. Okay, so what we do is a full 3-dimensional calculation, and then we just map the result onto this 1-dimensional path and analyze that. And the results are shown here. And what we see is that the function of time, we have this ballistic quantum behavior up to about 100 femtoseconds. But then there's a transition, essentially there's a localization regime, and we go over into a diffusive regime, which actually, if you look at it in more detail, is actually subdiffusive. So we conclude that there's no quantum speed up in FMO after about 70 femtoseconds, although the coherence still persists up to about 700 femtoseconds. So there's no quantum computation here. So why, now then a second issue is why is there no speed up? Well, it's really, if you look at it in detail, it's a combination of the fact you have localization due to the energy disorder in the system and also the effect of dephasing. And just as a reference system, you know, Anderson localization is very well known. Any variation in site energies in 1D will lead to localization, and this is a very strongly 1D system. Dephasing is somewhat more complicated. Dephasing allows the escape from localization initially, but overall it makes the transport subballistic. So the details of this are quite complicated, but we do understand it very well. Okay, that's the effects of decoherence. So that's actually known from the random walk. And maybe I'll just summarize here then. There's been many studies also from us and from other groups about the efficiency. And one of the figures that one sees often in this field is something like this, where the efficiency is shown as a function of dephasing rate. This is a pure dephasing model, so it's not terribly realistic. But it shows that there's a maximum in efficiency, and that reflects this maximum that we know from random walk theory that occurs at some finite value of the dephasing. And that maximum, it turns out for the realistic studies of this system for FMO, that maximum is right at the best estimate of the dephasing rate. So, and there's right now there's about 3 or 4 papers in the works on studying the optimality of FMO. And it does appear that FMO, the system, is optimized for the greatest possible efficiency of quantum transport in this open system. So that's one very good data point for understanding maybe the role of this. And let me skip this part. Okay, so I'm going to conclude here. So what we've seen is that there is entanglement in this system. And if we regard this in Schrödinger's language as the most remarkable and non-classical feature, it's really fascinating that it's exhibited in a biological structure. However, it is mode entanglement. It's a single excitation, and it is equivalent to the coherence. What is interesting, possibly more interesting, is to look at the larger light harvesting systems and see whether there is really true particle entanglement or the analog of particle entanglement, which is exciton-exciton entanglement. This is a much more complicated problem because generally exciton-excitons interactions are very strong and they cause annihilation. It's part of the photoprotection mechanism of these light harvesting systems. But still it remains to be seen. The second important, I think, contribution of theory is that there is no quantum speed up in the quantum information sense. So then coming to the biological implications, the efficient transport, it's clear that it's a very, very delicate interplay of coherent and decoherent dynamics. And the big open question is, what is the enabling feature or features? What precisely is the key thing which is responsible for the optimization of this? Is it the protein scaffold? Is it the pigment organization? Is it all of these things? And how can we pull this apart to understand whether or not the quantum coherence is actually important for the functioning of light harvesting? So that's also this second question here. What is the advantage conveyed by quantum transport to these systems? Is there a functional role? So that's still to be determined. And lastly, there's some future, actually really futuristic directions which we're pursuing under DARPA's view, which is to maybe apply quantum control methods to these systems. First, quantum control is very useful here to learn more about it, actually, because if you de-optimize a system, that may also, our hope is that will lead us to a better understanding of what is the precise part that is the most important optimization. And secondly, also to develop design rules for robust biomimetic systems, sensors, and energy sources as usual. Okay. Thank you. Oh, I forgot. I thought I should put up here. Schrödinger was obviously a wonderful personality in all respects, and he has in his book there's an epigraph for each chapter, and I think this epigraph really stuck in my mind for chapter 3. I think it's an inspiration for all of us that do theory. We try, was in schwankender Erscheinung schwebt, befestigt mit dauernden Gedanken. So we try to put down in lasting thoughts what's really just fluctuating up there.